Hello, everyone. Today we'll be discussing an article titled Synthesis of Membrane Proteins in Eukaryotic Cell-Free Systems. We initially narrowed our topic down to both cell-free protein synthesis and the synthesis of unnatural proteins. We chose cell-free protein synthesis because of its vast array of applications in biotechnology as a diagnostic tool and in medications such as insulin in diabetic patients. Also, unnatural protein synthesis can be achieved in cell-free protein synthesis systems, automatically eliminating this from our choices. Dr. Stefan Kubik is our corresponding author, and he received his PhD in molecular biology and physiology at the University of Stuttgart-Hohenheim in Germany. He is now currently the head of the Bioanalytics and Bioprocesses branch at Fraunhofer Institute for Cell Therapy and Immunology. Dr. Kubik has over 120 publications all in the area of cell-free systems. Some examples of his work are listed here. Dr. Sachesi is our first author, and she received her PhD in bioanalytics at the University of Potsdam in Germany. She then moved on to complete her postdoc in the Bioanalytics and Bioprocesses branch at the Fraunhofer Institute for Cell Therapy and Immunology. She is now the group manager in system software integration at Roche, which is a pharmaceutical company. Dr. Sachesi has 10 publications, all in the area of cell-free systems. Some of her work is listed here. So what is cell-free protein synthesis? Well, it involves the synthesis of natural and unnatural proteins in vitro and requires four main factors: cell lysate, a DNA mRNA template, energy factors, and post-translational modification processes. The researchers in this article decided to focus on eukaryotic membrane proteins because the molecular interactions of these proteins are still not fully known. Mutations in these proteins are also associated with heart dysfunction, migraine, and Alzheimer's. Now, challenges with this involve low protein quantity in vivo and tendency to form precipitates during extraction. There are some advantages of cell-free protein synthesis systems that address this, including increased protein output, preserved cell metabolic function in vivo, and reduced inclusion body formation. The first cell-free protein synthesis systems came around in the 1960s and included prokaryotic and eukaryotic batch operations. These systems only had about an hour of protein synthesis duration. Then continuous flow cell-free protein synthesis systems came along, and these utilized ultrafiltration membrane reactors and achieved 20 to 40 hours of in vitro protein synthesis. Subsequent studies focused on improving this system by optimizing the reactor pump, excluding air spaces from the reactor, and utilizing a hollow fiber membrane reactor. Studies after this decided to focus more on batch operations because of the simplicity and convenience of using these. Studies looked at optimizing ATP, GCP, NTP, and mRNA concentrations, increasing the rate of translation, including post-translational modification processes in vitro, and all of these improvements led to improved durations of protein synthesis of about 10 to 15 hours. The main competing technology is recombinant protein expression, which involves the transfection of foreign genes into the host cell. Applications are mainly in the pharmaceutical industry and the creation of vaccines and other protein-based treatments, including insulin for diabetic patients. Advantages of this technology include the fact that this technology can achieve industrial protein production quantities. However, there are several disadvantages, including altered host metabolic function, inclusion body formation, and the overexpression of the desired protein in the cell, which can lead to lysis.
How is this relevant to our course? This paper ties together cell signaling, cell differentiation, molecular pathways, and disease associated with unregulated cell growth. As we all know, proteins play an important role in the structure, function, and regulation of the tissues and organs, and thereby the overall function of the body. In this particular article, light is shed on the synthesis of membrane proteins, to name a few: EGFR, SERT, ZIP1. Therefore, an accurate model is required to study the action of various proteins and the resulting signaling cascade. Cell-free protein synthesis plays an excellent role for this because its performance does not depend on structural integrity of cells. As a bioengineer, it was fascinating to see the various post-translational modifications incorporated in a protein synthesized outside the cell. These systems allow one to directly control transcription, translation, and metabolism in an open source fashion. Additionally, this is also a very good model for proteins that are synthesized outside the cell, as I had mentioned, and also for discovery of drugs and for modeling drugs such as insulin. So, two major challenges were faced by these people when they were doing their experiment. First one would be the position of the membrane protein. Though the open nature of the cell-free system help optimize the environment for the protein structure and folding, it is tricky to place the protein in the membrane, which generally requires its translocation through a protein conducting channel. And hence they have used a signal sequence called melittin. The second one is the yield of the protein that they are getting. A higher yield of protein is required to better analyze the results that has been obtained, and hence codon optimization is used here. Codon optimization describes gene engineering approaches that use synonymous codon changes to increase protein production. It involves recombination based on different criteria without changing the sequence of the amino acid and can promote expression of the recombinant gene in different host organisms.
So getting into the paper discussion, in this study, the authors used cell lysates from an insect cell line to generate proteins using an in vitro cell-free system. They first wanted to test the potential for post-translational modifications in this cell-free system by glycosylating yellow fluorescent protein, or EYFP. They generated an mRNA template of EYFP with a melittin group that facilitates the insertion of proteins into microsomal membranes where this modification can occur. After running the reaction, they imaged the lysate mRNA solution and found EYFP localized to the microsomal vesicles. They then isolated their modified EYFP and exposed some of it to an enzyme which deglycosylates proteins. They separated the proteins using an SDS page gel and found those exposed to the enzyme had a lower molecular weight, indicating the glycosylated sugar molecules had been removed. Next, the researchers wanted to generate a protein with multiple transmembrane domains, as these are more difficult to derive from cell-based systems due to the incorporation of large quantities of foreign proteins into the membrane of the host cells. They chose a prokaryotic membrane complex composed of 3 subunits, Sec-Y, Sec-E, and Sec-G, with each subunit containing multiple transmembrane domains. They synthesized this protein complex using 2 methods. One involved 3 separate reactions for each subunit, and the other involved repeating the synthesis reaction several times in the same solution using different DNA templates for each subunit. The latter method resulted in all 3 subunits being present in the final reaction. Overall, the study shows the ability of insect lysates to generate proteins in vitro with or without transmembrane domains, as well as ones that can undergo necessary post-translational modifications. There are several limitations to the study that, if addressed, would better support the use of insect lysates as a preferred method for in vitro cell-free protein synthesis. First, no quantification of the protein gels were presented, and there was no indication of the number of samples tested for each experiment. Understanding the absolute rather than just the relative amount of protein synthesized would provide a better context for what this new method is able to achieve. Additionally, knowing the repeatability of the process is critical for assessing its future use. Another limitation of the study was that the researchers only showed glycosylation as a possible post-translational modification. To really understand the utility of the new method, it would have been nice to see other post-translational modifications like prenylation or phosphorylation to determine if the system could perform these functions as efficiently. Finally, there was no assessment of proper protein folding, a necessary feature if proteins are to be used for any functional assays. Only protein sizes were measured using SDS page gels. The researchers could have performed X-ray crystallography to map the structure of the synthesized protein and determine if the folding appeared correct. Alternatively, they could have performed a functional binding assay with other proteins, as correct protein folding would be required in order for proper binding to occur. So ultimately, the study shows that the potential of using insect cell lysates for in vitro cell-free synthesis, but the new system's efficiency, utility, and accuracy need further investigation before it can be adopted for broader use. So here are our sources, and we thank you all for listening.